This is Nanoscape. I'm Seth Zimmerman.
And I'm Erin Spain.
On this show, we venture into the frontier of nanotechnology
With experts in the field who are turning the science of small into the next industrial revolution.
This podcast is brought to you by the International Institute for Nanotechnology at Northwestern University.
Our guest today, Professor Julius Lucks, is working at the intersection of two very exciting fields, synthetic biology and, of course, nanotechnology. His research focuses on RNA, tiny molecules that can sense and respond to their environment, and he's using them to build tools that tackle global challenges in human health and water quality and the environment.
Julius is the Marjorie Claire Carlson Professor of Chemical and Biological Engineering at Northwestern University and co-director of the Center for Synthetic Biology at Northwestern. We welcome him to talk about how nanoscale thinking underpins many of his breakthroughs and how he keeps people and real-world needs at the center of his breakthrough research. Welcome to the show.
Thanks for having me.
Wonderful to have you here.
So I love this. You trained as a chemist and a chemical physicist and then became a leader in synthetic biology. Quite a change, it seems like. So looking back, what drew you towards studying RNA as a dynamic system? And when did you realize this was where you wanted to focus your career?
Well, life is an adventure and it takes you in different places. I grew up on the Outer Banks of North Carolina, normally known for its kind of surf culture and whatnot. But I was one of the kids that instead of surfing, I did math competitions for my fun. And actually, I wanted to be a mathematician early on, but you explore different things in life. I came across the wild world of chemistry and I was like, that's really cool. This is really interesting. I actually started out in organic chemistry, trying to synthesize molecules. But then I was like, I don't know, I'm kind of missing the math piece of this. So that got me into physical chemistry, eventually physics. And it was actually in my PhD in physics. I was trying to find a PhD advisor because I didn't have a physics background. A lot of the different professors basically told me that I wasn't smart enough to do their research. So it was a little bit of a challenge there, but you persevere. And actually I came across a professor who was eventually my advisor that was starting to transition himself from really classic physics into biophysics. And I did well in his classes and we were talking, and apparently to my advantage, physicists are afraid of molecules. But I had a chemistry background. I wasn't afraid of thinking about molecules. And so he's like, okay, let's explore this biophysics thing together. And I thought, hey, this would be cool. I can kind of go where I want. But he plopped this one paper in front of me, was about how to calculate the shape of an RNA molecule. And I was like, all right, I guess I'm doing this.
What was in that paper that hooked you?
RNA molecules are so wild and beautiful. They're all throughout our body. They outnumber the proteins and the DNAs, and they exist in this nanoscale world. But it's a very dynamic world where these molecules are wiggling and jiggling and changing their shapes and all this stuff. So it's a really cool problem that's related to how these molecules work in life and how you might engineer them to try to understand their shape. So I started calculating and doing all that stuff in physics, but as I got further along, I got more interested in the biology than the physics. And so when I transitioned to a postdoc, I had a chance at UBerkeley in the Miller Institute to just try something new. And I was like, hey, I'm just going to be an experimentalist now. And I wound up in a lab that was doing synthetic biology. And I was like, hey, this is really cool. It's like a way to do science through engineering, try to build something and it doesn't work. And then you got to understand why. And that kind of defines your next scientific quest. And it's been an adventure ever since.
I want to go back to the RNAs a little bit because you mentioned they're pretty powerful to work with, even though they're so tiny. So talk to me about that. What makes them such a powerful material to work with?
Well, I like to think that RNA is life's magic molecule. It's really special. You hear about the DNA genome. It's like nature's hard drive. It's got all the instructions and stuff. But, DNA by and large, it stores information in all of our bodies, but it doesn't do much else. And there's a reason for that. It's because it's really good at storing stuff and not doing too much else unless you engineer it on purpose. But in natural biology, it just kind of sits there. RNAs, they have wonderful properties like DNA. They store that genetic information. But because they can change their shapes and move around, they do a lot of things in the cell. And just how evolution is kind of worked its way out, RNA is the only molecule that can store and propagate information like DNA, but then do stuff like proteins. And so it sits right at the crux of a lot of biology. So RNAs are the ones that are determining kind of when genes are made and how much genes are made. They can even do chemical reactions. Like it's the RNA molecules that make proteins. So from a basic biology standpoint, they just sit right there. And that makes them so intriguing to think about how to engineer into biotechnologies because they do so much.
I think everyone's going to have a visual what a DNA and a DNA strand looks like, and they probably don't have the same idea of what RNA looks like or how to visualize it. So an analogy I've heard about for RNA folding is that it's like origami. Is that the best? Can you explain that analogy, or is there a better one?
So if I took the origami analogy, I'd have to adapt it and say, if you could make origami out of Jell-O and put it on a giant shaking machine that's constantly jiggling around, that's probably more accurate. I mean, I think a lot about like a spaghetti molecule that's like kind of sticking on itself and flying around. I mean, they're absolutely wild molecules. You know, if you got an image of a DNA in your mind, then it's not too far to imagine that thing just kind of wiggling around. There's different shapes involved. They're absolutely beautiful and intricate wiggling snowflakes, if you will. They're all different. I will tell you a fun little anecdote. Was a project we did to try to make a movie of an RNA molecule that's being made in the cell. They're made kind of one base at a time in a line. But as soon as they're made, they start to fold into shapes and they wiggle around and they change their shapes. And so we invented this kind of new experiment to use next-gen sequencing, like a chemical microscope, to get this data. And then we had these algorithms that could process the data. Anyway, we made a movie of an RNA molecule that's being made in the cell and published a paper, and then we tweeted the movie out, and it was like getting some hits and stuff. But then someone downloaded the movie and re-uploaded it, but put the dancing RNA molecule to salsa music, and it went viral.
Cool.
People were
cool.
like, "This is so cool! I understand RNA now." And then it got picked up by Francis Collins, who was directing the NIH at the time.
Wow.
I was like, "Hey, saw this salsa?
Get me that
RNA.
guy who did that.
Can I?" Yeah, exactly. He's like, "Can I do a blog post about your paper?" And I was like, "Absolutely." And then it was super awesome because we got this email exchange. He's a big folk musician and I play mandolin. And so we were like going back and forth, like, what's our favorite bluegrass tunes? So, hey, this is where dancing RNAs can take you.
I love that in so many ways. And I wish I could talk more about the mandolin because I'm trying to put together an IIN band or some sort of side
Like a performance?
Let's do it.
performance or
Yeah.
a talent show.
Talent
I
show
love it.
is a great idea.
Yeah.
I'm down. Sign me up.
Much of your work starts with fundamental questions about RNA folding and dynamics and ends with tools that work in the real world. How do you decide when a discovery is ready to move from basic science into something that's applied?
It's kind of a cycle. It's not really a linear process. And the way that we have kind of bridged these fundamental to the applied has been a little bit, again, of an adventure and lots of wonderful luck and circumstance. We happen to be thinking a lot about RNAs because they're really important in biology. They're related to disease. A lot of diseases are RNA diseases, and so the treatments can use RNAs and all these things. But still, you need to understand these shapes and how they're wiggling and jiggling around. And so we were exploring that for a while, and we got really interested in a certain type of RNA that actually helps cells sense and process information. Well, I think this is one of the coolest things about biology. Cells are out in the world and things are changing and they're adapting and they're kind of like making decisions with the information that they receive. Like the temperature changes and they decide, oh, I'm going to do something else. Or they see a food source and they need to figure out how to eat it. But they kind of make these decisions without brains. It's like, whoa, what's going on? And these simple cells, it's a lot of that processing is because the RNA molecules are doing it. You can almost think of them as like computers, that like molecular computers, except they don't do zeros and ones and social media posts. They like bind chemicals in the environment and they trigger a gene to turn on and so on and so forth.
This understanding eventually led to a collaboration with Sarah Young, who is an anthropologist here at Northwestern, and also your wife. Tell me about that.
What happened was Sarah, who's in the global health space, is thinking about water contaminants all the time. And we were talking one time and she was like, hey, I found this use case. Wouldn't it be great if you could give a technology? This sounds kind of random, but this is how it works. You give a technology to people that could tell them if their water is safe to drink. Like, wouldn't that be cool? And I was like, that would be cool. I don't know how you'd ever do that. But then I was like, okay, this is interesting. I hadn't thought about water before. Let me Google it. And Googled like what's in water. And it turned out that there is some contaminants in water that are contaminants to us are also contaminants to cells. And it's like, okay, that's kind of interesting. And so these toxic compounds, cells are trying to sense and then do something about it. And it turns out they're using the RNAs to do it. And so I'm like, whoa, we're sitting on a potentially cool way to deliver this. So what we did is we used all of our knowledge about how these natural systems worked. And then we used some really cool tricks in synthetic biology. We kind of took them out of the cell, reconfigured them at the molecular level to sense these contaminants and then basically glow. So people could add a drop of water to this test and it would light up if it's bad, if something's bad in there. So I mean, you can think of it like an at-home COVID test, but not for COVID, for like a water contaminant, something like that. And it was the first in my own experience of kind of going coast to coast where you've got that fundamental discovery that translates into the application. And we've taken this to Africa and tested it out. We're doing it in Chicago right now and really giving people these tests that have these cool program molecules. And now it's a little bit of like I said, it's this cycle. Like people are like, oh, well, can you test for that or something new? And it's like, oh, well, we can't. Now we need to go ask a scientific question and answer that question. And so we're almost letting the technology drive the fundamental science now.
Well, and this technology, this is called Rosalind, right?
That's right, yeah.
That's right, named after
Rosalind Franklin, that's right.
And Rosalind Franklin was?
Rosalind Franklin was a key scientist in the discovery of the DNA double helix and how it works. So everybody quotes Watson and Crick for determining that double helix structure, but it was really Rosalind Franklin's data that they based their interpretation on, and she's very much in the mix. And I think it's unfortunate that she wasn't recognized at the time, but it is really nice to see people realizing the importance of her contributions. And so you got to have a great acronym for any technology.
Right. You got it.
And we were brainstorming some ideas in my lab at the time, working on the technology, came up with that acronym, and I was like,
beautiful.
Done.
Done.
Yeah.
Well, something else I want to mention, it is pretty cool that you get to do this kind of research with your wife.
Yeah. So, it's amazing what you can do over dinner.
Right.
If you're both in the same area. It was interesting because she brought that challenge to us right when we were both moving from Cornell to Northwestern to be a part of the creation of the Center for SynBio. And I was thinking at that time, wouldn't it be great to kind of do more than just the fundamental stuff, really try to touch people's lives? So it's a perfect context.
I mean, I just want to dig into that a little bit more. Like you said, you're at the dinner table talking about your work, and then you're out in the field, like in Africa, testing these technologies.
She tells the story, when our kids are not interrupting us, we can talk science. It's actually, it's been a beautiful thing to not only do it in terms of being life partners together, but like scientific partners, because she's coming at the problem completely differently. She's interacting with the people, the stakeholders, kind of studying at that human level what people need. And we're in the molecular world, on the nano, we're in the nano world over here. It's been incredibly eye-opening for us, really kind of defining what scientific impact means, what we're trying to achieve. Getting to go in the field, obviously you're going to be able to road test your technology and see if it works, and that's great, and that's super needed. But you get this human element. You meet the people that you're really trying to help, and that gives you a couple things. One is like, you really hear from them what they need. You don't just kind of make up this thing in your mind, like, I'm going to make this thing and people are going to love it. Well, actually, they didn't want that. They wanted something else. So you get to hear from them first. Then you also get to see in every case we've done it, just like how important this type of stuff is to everyday people. They see it on the ground. They see what you're trying to do, and you're trying to help together, do the together. We're not coming in white lab coats to save the day. We're like, no, we're here to try to work with you to do it. It's super inspiring. And I try to take as many lab members and students out into the field as I can to give them that context. And that anthropological perspective is absolutely critical to do that because, we spend a lot of our time dreaming at the nanoscale or in the lab, and knowing how to interact with other people, how to embed yourself in other cultures and respect and learn about other cultures, and even how to incorporate feedback kind of formally using social science techniques to interview and process that data. It's something that lab scientists, we're not trained in, and it's very hard. It's very challenging, but it's super rewarding, and it really makes the work a lot more rich.
I think a big thing that we've learned from doing this podcast is how important interdisciplinary collaboration is for work in the nano field. And I'm always curious, when collaborations fail, what actually usually breaks first? Is it the science, the communication, or is it the incentives? Where do things go right, and when do things go wrong?
I think collaborations cannot go right for a number of reasons. Sometimes you've got that amazing idea on the whiteboard, and nature's just just not going to let you do it. It's just not going to work. That happens plenty of times. Or maybe you're not aligned on goals or something like that, and the team's not working together effectively. I think to make it work, you got to really invest. And, multidisciplinary work is super impactful, super important, but it's also really hard because everybody's coming from a different perspective. That's what makes it magic. But really having some glue between everybody's aligned and everybody can understand each other. Even within a project team, sometimes you feel like you're speaking a different language, different scientific language. I've been really grateful for Sarah's perspective and leadership in this. Now, we kind of started small, working just with Sarah, doing some stuff, trying a little field trial, didn't know what we were doing it in the US. So that's a little bit easier. As we built up, she added more of her anthropology team to it. And now in the context of this Chicago project, what was great is because we got this NSF grant actually required us to make a community advisory board, which I'd never done. So we're in Southside Chicago and Evanston. We've got community members. These are people that just live in these communities that are plugged in. We've got older people. We've got kind of environmental activists. We've got water experts and whatnot. And it's this hugely different perspective, not only on the challenge, but how to interact with these communities. There might be some skepticism about the university and what it does and like, how's it going to help me? We have community members that are kind of the leading edge of working with these people. Like, no, that's actually really great. You should come by. You should get to know them. When you put that effort in to really build that collaborative team and bond and get aligned, it can be incredibly powerful.
And is the device being used in the Chicago project to test for like lead and other contaminants?
Yeah, we're focusing on lead in Chicago because it's such a big issue. And there's some other interesting federal initiatives in the Biden infrastructure bill. There was quite a bit of money to remove lead pipes. And there were some changes to the EPA regulations on what the toxic levels of lead are in water and stuff like that. So kind of now is the time to address the lead problem. Although we are very broadly interested in lots of different challenges that are out there. In fact, we're just invited to be part of a multi-team project to go beyond water and detect food quality.
Why is this early detection such a game changer across so many fields? And why is nanotechnology such a great match for this problem of making the invisible visible?
We've been living in the quote unquote information age for a long time now. And we consume so much information. We generate so much information. And now AI is being trained on this and it's even more powerful. But if you think about the information that we get, there's not a lot about our environment, our physical world. We've got some weather predictions. We've got some temperature stuff and some humidity stuff. But there's this whole world out there of chemical signals and nutrients that we need to consume and toxins we need to get away from and things in the air, things in the water that, you want to avoid. And so we actually are lacking a lot of deep information about the things that are pretty critical to our health, the health of our environment. You try to think about, okay, how can we access that? Well, you need technologies that can measure these things. And it's not like they don't exist. There's some great pieces of equipment that's about the size of my minivan that will do this for you. It's very expensive, sits in a lab, you need some expertise. So if you really want to gather that information at the scale that it becomes useful, and that scale is really large, it's like on the scale of billions of people and all the places that we live, then you need to find a different way to do it. And nanotechnology is the perfect solution because all of the things that we care about sensing at this scale are nanoscale things. And for our particular interest, because biology and cells and microbes and plants are already sensing these things, those nanotechnology mechanisms that they use are the perfect starting point for this.
So about that, another project of yours, Plant DX, it aims to help farmers detect crop disease before visible symptoms appears. How does the nanoscale biological sensing change the way we think about sustainability and resource use in agriculture?
Yeah, this is really interesting. So this was a project that was originally designed to try to empower small stakeholder farmers with information. The majority of the people on the planet are small stakeholder farmers. These are people that are growing enough food for their families, maybe a little bit extra to sell at the market, but they're not large food producers. They're not going to the grocery stores and stuff like that. They're growing their own food. So you can imagine how important farming is to their lives. And in certain parts of the world, I mean, pretty much everywhere, crop pathogens, viruses, fungus, and stuff like that can be really devastating if they get into your farm because they infect one plant and they hop to the next and whatnot. In the milder cases, they'll like reduce your yield or make your potatoes smaller or something. In the worst cases, they'll just kill your crops. And as with a lot of these types of things, if you're seeing it on the leaves, you're seeing some brown spots, you're seeing that stuff, it's way too late. Like you've got the infection and it's probably spread. So the idea was like, hey, can we make it a way for people to test their crops early before you see any signs and give them information about what they can do about it? Maybe if they find a region of their plot that's got this disease, they can take those plants out or do a local burn in that area and stop the spread. Or a lot of times in these communities, people will share seeds and stuff like that. And if you find it on one farm, you say, okay, don't share anything from this farm, just kind of lock it down. So this was a really interesting idea because we had played around with this concept prior to COVID, and we're doing some nano engineering to try to figure out how to detect these viruses. Interestingly, a lot of these viruses are what are called RNA viruses, meaning they use RNA in their genome, not DNA. So it's kind of a natural fit for what we're interested in. Yeah, totally.
Yes.
And we were exploring it. Then COVID hit. And of course, everybody had to work on a COVID project. So we were like, hey, can we kind of do the same type of thing, but for COVID? And we stumbled on this great work being done by a bunch of others to use these CRISPR mechanisms. So the CRISPR, the genome editing tool, can be programmed as a diagnostic tool for these viruses. So we were putting that together and we're like, hey, nobody's doing this for crops. So that's what we started to do, put it together, and a really great project. We took this one out to Kenya to tested the technology, interacted with the farmers. And this is where that human connection became really critical, because what we actually found was really cool technology, but probably not the best for the farmer. Maybe it's a little bit too complicated for the target farmer. And also we're realizing that farmers have a really kind of almost like a familial relationship with their crops. They're so precious to them. So if they find something wrong with a crop, they're not likely to take that crop out. They're likely to just say, I'm just going to pray and hope this thing works. But what we did find is that upstream of these farmers, where the seed producers they're selling the seed, they have to do a lot of testing, and they're looking for technologies that they can do cheaper and faster and whatever. So it's really the seed producers, the upstream, target the upstreams and deliver the farmers clean seeds to begin with. So it's still going to have an impact, but not the one that we originally thought. And we would not have learned that if we weren't out in the field talking to farmers ourselves.
I mean, could you have imagined at the beginning of your career that you would be out in a field in Kenya, Africa, doing this type of work when you were studying RNAs? And
Absolutely not. It has been just like a twist and turn and an adventure, and it's not come without its challenges. Back in the days when I was just doing academics, to switch from chemistry to physics wasn't easy. But, I kind of got almost addicted to learning hard things. And I learned that if I can go into a new space that I don't know anything about, then at the end of every day, I know 100% more than in the morning. And that's just like a really cool feeling.
Yeah.
And so this is kind of the next level of that. And who knows where, I'd probably end up doing something else in 10 years or so.
You've been very involved in shaping synthetic biology as a discipline, including being the co-director of the Center for Synthetic Biology at Northwestern. From your perspective, where is the field right now compared to when you started, and where do you see it going?
Yeah, so I got in the field, synthetic biology, 2008. I think many people would put the founding around 2000. And the synthetic biology is really like an exploration of what you can do when you engineer biological systems. It was actually kind of founded by some physicists that were thinking about really interesting things that cells do. They form patterns, they oscillate, they have molecular clocks and things like that. And they're really interesting physics concepts. And so what they were doing is they were taking molecules in nature that do something else and kind of wiring them up to do these cool physics things. And we're having some success. It wasn't trivial, but they were showing that if you take natural molecules and you wire them differently, you can make them do different things. And so people got this idea like, wow, nature does so many cool things. What can we build out of them? And the field at that time was very much in this exploratory phase, trying to merge biology, but with physics and chemistry and computer science and just engineering techniques. It was almost trying to say, let's turn genetic engineering or biological engineering into a true engineering discipline, not just in name only. And so the field was full of a lot of promise and just a ton of excitement. Like anything you would do is going to be amazing, but still very early on and still really hard to do even the basic stuff, like make a cell, make make this compound or something like that, which sounds easy, but it's actually kind of tricky. So over the years, I think the field has matured. It's gotten a lot better at some of the basic processes and building infrastructure. Speaking about nanotech, a lot of nanotech involves DNA. Same thing in synthetic biology. DNA is nature's instruction set. At some point, you're going to make some synthetic DNA to control this biological system. And so we've gotten really good at synthesizing DNA and combining DNA in different ways. We've gotten really good at predicting the shapes of molecules and how to design certain types of molecules. But some things remain challenging. It still remains, we can't build a cell from scratch right now. So there's a lot of really cool intellectual challenges. I mean, as we've progressed in our capabilities, we're starting to see the fruits of our labors in technologies that are really starting to make a difference in the world. So we talked about CRISPR earlier. That's a synthetic biology tool, such a programmable molecule. And the fact that a few months ago, a baby was cured of a rare genetic disease in like a month by programming CRISPR, that's amazing.
Yeah.
So that's coming online. Things like mRNA therapies, programming how you want your body's immune system to respond to COVID or to cancer at the nano level. That's kind of amazing. That's a whole new way to think about how to make medicines that's coming online.
And then you throw AI into it, and now it's opening a whole new door.
Totally. And so that is kind of where the field is going. For a long time, the field was rooted in physics. And so ultimately, we want to be able to design biological components into these technologies. And a lot of those methods were kind of rooted in physics and biophysics and a lot of great stuff there. Now with AI, we have the ability to synthesize this large amount of data that we have to try to make the next iterations of design algorithms. And in some spaces, like in the nanospace designing proteins, that is like Nobel Prize a couple years ago. It's really going into a ton of different technologies. That is happening, and it's exciting to see where it's going to go into designing some of these more complex systems that can do even more cool things for humanity.
As we're thinking about synthetic biology, nanotechnology, the sort of human interaction that you're able to have using some of these technologies and AI, what excites you the most as we look forward to the next decade of research and the new tools that might ultimately be used?
I am most excited by developing computer-aided design tools, CAD tools for biological systems. If you look at almost any true engineering discipline, they've got a set of like software tools that can help them design it. So I like to, when I teach these subjects, I go to this company called Autodesk, which makes a lot of these tools. And so if you're a mechanical engineer, you can go to Autodesk and you can find a tool that'll design you a bolt all the way up to a city. So if you're a mechanical civil engineer, you've got a set of tools that help you think through the details of designs. If you are an electrical engineer, you've got tools that can help you think about how to design a transistor, lay them out on chips, all the way to designing laptops, right? This is why these industries are so powerful. What can we do with computers? Almost anything. And the price is dropping and more people access them and everything. So if we really want to change the world with biology, which I think we totally should for a lot of great reasons, then we need to get there. We need to have these tools to help us deal with the complexity of biology. We need to have these tools to help developing the next synthetic biology technology cheaper and faster. There's a lot of challenges in the world, and we just don't have time for it to take 10 years and $1 billion for every one of those. We need to really get rapid about this and develop infrastructure to support manufacturing these types of technologies, distributing these types of technologies.
So what about the next 10 years? Where do you see the most impact happening in the next decade?
So I'm looking at the next 10 years as scientific breakthroughs in our ability to design biological systems beyond just molecules, not just individual molecules, but collections of molecules, maybe even whole cells, things like that we need for these applications, getting the applications to the point where they're useful to society. People can use them. They can use them safely. They're helping people's lives, and kind of building that infrastructure so that the next technology that comes down the line comes in half the time and half the cost.
All of this comes back to like making this a better world, really, in a
Absolutely.
lot of ways. That's kind of the goal.
It's absolutely the goal. And the way I view it is why synthetic biology, why nanotechnology? Humans are biological systems. A lot of our natural world is filled with biological organisms, the trees, the animals, all that stuff that we all need to coexist and we all need to sustain each other. And so technologies that leverage the capabilities of the natural world. It does so many things. But since we have to interface these technologies with biological things, that's the best technology that you can use. There's lots of other things. I'm not going to cut out all of my other colleagues that work on amazing technologies, but I think there's a lot of great things you can do with synthetic biology. And I'm also not saying that technologies have to be purely biological, right? But having biological components, do what biology does best. There's really cool projects happening at Northwestern, Jonathan Rivney's lab, things like this, where it's like, all right, I want to make a device that goes in your body and delivers medicine on time. Well, there's a device component to that because you need the electronic communication and the interface there. So biology is probably not going to do that well. But can biology make a drug on demand? Absolutely. So that's the perfect type of interface I think we're going to see a lot of.
I want to ask about the Lucks laboratory or laboratory, depending on how you want to pronounce it.
I'm an Anglophile, so laboratory works for me.
So working with the next generation of scientists is important to you. And I'm curious, when you're leading this next generation of scientists in RNA and synthetic biology, what is the most common early trap for talented students, and how do you help them climb out of it?
That's a really great question. We'll contextualize that a little bit. I love that we're developing technologies and we're trying to do our part for the world, but I do think as a laboratory, our most important contribution is training the people that come through the lab. And this is a real privilege of being able to be in a university and have that training and education mission as well, because these people are amazing, first of all. They're going to spend some formative years here at Northwestern, and then they're going to go off out in the world and do amazing things. So it really amplifies everything that we're trying to do. What I see, though, people come in, they're absolutely incredibly talented, and they want to change the world. And that's great. I love that huge drive and ambition. And I see a major challenge, though, is that changing the world takes time and it's hard to do. And oftentimes you need a lot of other people to do it. So a common pitfall I see students go in is they get really excited about that project that is it's the grand slam. And that's really cool. But what I try to do is I try to take that vision of theirs and help them hit the single first. Get on base. Take a little piece of that because there's so much to learn. People come here, they've got great backgrounds. There's still a ton of things to learn. Synthetic biology is still a really new field, so there's a lot of catching up to do. We're even creating new ways to teach synthetic biology here at Northwestern, so we're grappling with these challenges. There's a lot of learning, and I like to get a piece of that vision so that they're excited about it, but they can still learn all the things that they need to do. And then as a student, there's nothing like the thrill of a discovery and the confidence that it gives you to really go after the challenge, because some of these big things, they're not going to be linear paths. Your first ideas are not going to work. It's going to be a really hard slog through some hard times. And I think that confidence is really important to help you be true to yourself and know that you can do it somehow. So I try to help them carve it out and pump them up and get them through.
So you never tell them they're not smart enough, like that other...
I haven't, I have not passed on that part of my training.
That gem has not carried on. Okay.
I have not passed on that part of my training.
That gem has not carried on. Okay.
Dr. Julius Lucks, thank you so much for coming on the show and explaining this incredible research that you're doing that's really touching the lives of people all over the world. Thank you so much.
It was really great to be here. Thanks for having me.
Thanks so much for listening. This podcast is brought to you by the International Institute for Nanotechnology at Northwestern University. Be sure to follow this show on Apple Podcasts or wherever you listen to podcasts, and be sure to give us a 5-star review.
For more information about IIN, go to our website, iinano.org/podcast.